Thank Hu Lin and his colleagues for hosting a really wonderful summer school and symposium, and Jean and Debbie for making it all run so smoothly. I'm going to talk about affinity maturation by somatic hypermutation and selection. And for me, this is a return to my beginnings as a computational immunologist. The first time I was exposed to computational immunology, I wasn't quite sure what it was all about, and I began to look at immunology and try to determine whether or not it was something that I wanted to do. And it seemed fascinating. Then I got to the chapter on somatic mutation. And you may know that somatic mutation occurs by the introduction of point mutations into immunoglobulins and their subsequent selection. And I was astonished. I was a physicist at the time and was fascinated by Darwinian evolution. And the idea that this process was going on every minute of every day within our bodies was astonishing. And I thought, how could I have lived for 23 years without knowing about this? It was just amazing. And so my very first paper or two in computational immunology was about affinity maturation, and I finally come back to it again after 20 years or so. And here's the man who introduced me to it. Before I get started, though, I want to put a plug in for next year's summer school and symposium. Next year is going to be in Boston, and it's going to be co-hosted by the Hariri Institute for Computing and Computational Science and Engineering. It's a brand new, beautiful institute, and it'll be a very nice venue. I want also to let everyone know that because I have just moved from Duke to BU, I have several positions that I'm trying to fill. And in particular, postdocs and staff programmers. If anybody out there is desperate for a job in these trying economic times, you might want to consider lovely Boston. This is overlooking the South End where the Boston University School of Medicine is located. And then, of course, there's also beautiful Fenway Park. And please consider it. As Rob did, I'd like to make sure that I don't neglect my acknowledgements. So the work that I'm going to describe today is really occurring in the intersection of two large programs. One of them is a program that I direct, and it's the development of software for the analysis of affinity maturation and the germinal center response. And the people who are involved with that are the people at BU that I'm showing here, more people at Duke who are remaining with the project, and the experimentalists on the right, including Jason Hawk, whom you're going to hear from a little bit later on, and Haberman at Yale, and Phil Hanna at Michigan. And the other project is a project for the discovery of HIV vaccines, which is a very large project headed up by Bart Haynes. And I'm going to start off by talking mostly about that work, and that involves mostly Bart Haynes, Garnet Kelso, and Steve Harrison, who with me have been working on these particular problems in the context of HIV, although we'll also be talking about influenza. So what I would like to try to get through is first to introduce you to affinity maturation and somatic mutation in a little bit more detail. Then I'm going to describe to you the methods, the computational, mathematical, statistical methods that we've developed in order to study these processes. And then I'll present to you the results. The results will be on a dissection of the clonal history of an influenza infection and a similar dissection of clonal history in an HIV infection. And the lessons that we take from the HIV case will then motivate us to consider a new method for designing vaccine immunogens, and I'll describe that. And then if there's any time, and you haven't all abandoned me for coffee and donuts, I'll switch gears and talk about the modeling of the germinal center reaction that is largely the province of the other group. So let's remember our antibody structures and genetics. So remember that T cell receptors and immunoglobulins are constructed through a process of recombination, right? And to get an immunoglobulin heavy chain, one chooses a V gene segment, a D gene segment, and a J gene segment, and cuts out the DNA intervening and ligates them together. So we have a great deal of diversity by recombination. Furthermore, we have diversity introduced through the choice of the specific place that the DNA is cut, and from the addition of non-templated or N nucleotides in the junction as well. So combinatorial diversity, diversity in the location of the recombination site, and diversity in the addition of N nucleotides. Now, the reason I'm going through all of this in such detail is that the task that we are going to put to ourselves is inferring what these processes were for any given clone of antibodies that we isolate. Now, once we have gotten our antibody genes through these several processes prior to antigen exposure, we then find that there is further diversification after antigen exposure. And the first hints to this came in 1964, a very important paper by Eisen and Siskind. Eisen and Siskind had just developed a new technique for the measurement of affinity in serum antibody. And they were testing it out on immunized rabbits. And what they found was that, and I've just picked out one row here so that you're not overwhelmed by the large number of numbers. After 2 weeks, the affinity of the serum antibody for the antigen was 1 microliter per mole. And after 5 weeks, it had risen by a factor of 6. And after 8 weeks, the affinity was now 250 times higher than it was initially. So a really spectacular increase in affinity in the serum antibody. And this, of course, came to be known as affinity maturation. And we now recognize that affinity maturation is an absolutely essential process, that it is this process that provides us with the high affinity, high specificity antibodies that are borne by the memory cells and long-lived plasma cells that provide protection against the second encounter in a natural infection or against the first encounter in a vaccine. And so what we are trying to do when we develop humoral immunity-based vaccines is to induce a potent and effective affinity maturation. So 20 years after Eisen and Siskind had shown that there is such a thing as affinity maturation, Martin Weigert and his colleagues, studying the kappa light chain in mice, made the observation that they could take the kappa genes that they induced by immunization in a mouse and derive them all from a single germline version of a kappa chain by introducing point mutations, and they would get a nice tree structure like this, and it led them to understand for the first time that somatic mutation was going on. And they put 2 and 2 together and conjectured that it was this somatic mutation that underlie affinity maturation. And that was subsequently confirmed by a number of different people, and so the story is now really very clear. What happens is B cells that are elicited by an antigen enter a process by which point mutations are introduced into the antibody genes. The antibody genes that by chance encode antibody molecules with higher affinity for the antigen provide a proliferative advantage to the B cells, which then go on to eventually dominate the humoral response. This is an example of affinity maturation shown by our colleagues at Duke. And what we're seeing here is the effect of a vaccine adjuvant on affinity maturation. This is the log of the relative affinity, and this is mice immunized with recombinant protective antigen from Bacillus anthracis, the agent responsible for anthrax. And in red, we see, I'm putting my finger in front of the laser. It really doesn't work at all well that way. In red, this is immunization with alum as the adjuvant. Alum is the adjuvant that's typically used in human vaccines. And in green, it's a vaccination with no adjuvant, with just the antigen. And we see that there's a substantially increased affinity maturation in the presence of an adjuvant. And so that's really what our group is interested in doing, is trying to determine which adjuvants provide us with the best affinity maturation. Why do we care about affinity maturation? This is another piece of data from our colleagues at Duke. What they've done is they've looked at the affinity and the neutralization capacity. So they take serum and from different points in the mouse experiments, and they measure the mean affinity and the mean dilution, the mean neutralization capacity by seeing how well the serum inhibits the killing of macrophages by anthrax toxin. And there is a very nice correlation. The higher the affinity, the better the neutralization. And we are also now with Phil Hanna conducting actual survival studies, and we expect, of course, that we'll see something similar. So what we're going to want to do is to study affinity maturation. And what that's going to require is that we isolate antibodies that we get during the course of affinity maturation, and then revert them to their ancestral state. Because we won't have the opportunity to isolate antibodies at different points along time because there won't be enough to begin with. So what we do is we isolate antibodies near the end of the process and then infer what they looked like before the mutations were put in. And so that's really what I want to describe right now is the statistical methods behind this. And I recognize that statistics is often not terribly exciting. Apologies, Bets. But this is what I spend a lot of time doing, and so now you're going to have to listen to it. And then we're going to get to the fun part after that, I guarantee. So what do we need to do to infer the ancestry? Well, it's actually very straightforward. I mean, if you think about it as a statistician would, there are parameters that you have to estimate. What are the parameters? Well, you have to decide what is the appropriate VH gene segment. That's just a parameter. It's one out of the 48 that you have available to you. You have to decide what is the recombination point at the 3' end of V. That's just an integer. You have to decide what the N nucleotide sequence is between V and, oh, that should say D, I apologize, between V and D. And so, you know, we've got these 4 recombination points. We've got 2 relatively short DNA sequences, and then categorical variables for V, D, and J. Straightforward. Now, the thing that makes it interesting, though, is that because we are allowing somatic mutation to intervene between the original rearranged ancestor and the observed antibody, any antibody gene can give rise to any other through the introduction of point mutations, insertions, and deletions. Well, of course, which ones do we choose? We choose the ones that typically involve the smaller numbers of mutations. But why is that? That's because, I beg your pardon. That's because we understand that mutations are relatively rare, and we are going to use a model for the introduction of mutations by which the probability that nucleotide state A gives rise to nucleotide state D over a branch length L is given. Now, before we get to that, let me go back and provide an illustration of why this is something that actually matters. So VRC01 is an antibody that was isolated by the Vaccine Research Center at the NIH, and it's a very important antibody. It's very important because it turns out to be a broadly neutralizing antibody against HIV. And we're going to come back to talk in more detail about these broadly neutralizing antibodies. But the point is that it is capable of neutralizing a large number of different strains of HIV-1. And what this means is that in contrast to other anti-HIV antibodies, HIV will have a very difficult time escaping from a humoral response dominated by VRC01. Because whereas an antibody that, a more typical neutralizing antibody, which binds to the hypervariable loops on the HIV envelope protein, the HIV envelope is highly mutable, and as soon as there's any pressure on the HIV, it can mutate and thereby escape. VRC01 is directed against a highly conserved part of the HIV, and it is very difficult for HIV to escape from it. So there's been a lot of attention paid to VRC01, and it's also, it turns out, 32% mutated. 32% of the nucleotides are not what they were when it was first rearranged. This is an extraordinarily high level of mutation. And so it seems clear that affinity maturation has something to do with the properties of this antibody. And so we are very interested indeed in understanding exactly what happened to this antibody during its life. So what we want to do is revert it. Let's take out the mutations and see what the pre-mutation rearrangement looks like. And by the way, this is what the discoverers did in this paper as well. John Mascola is the senior person there. So let's take a look at an alignment of VRC01 with the gene segments that are the most likely participants in the rearrangement process. So this is VRC01 up here, and I'm showing the invariant cysteine codon TGT here, and the invariant tryptophan codon TGG here, and this is CDR3, complementarity determining region 3, which encodes the large CDR3 antigen recognition loop. And this is VH1-2, and matches between the top and subsequent lines are indicated by a dot. And so VH1-2 matches up to here, then there's a mismatch. 2 more matches, mismatch. 3 matches, mismatch. And so the question is, where is the recombination site? Where did this end and N nucleotides begin? Because it's important. So, first of all, this almost certainly is encoded by VH. So, you know, maybe this is the first nucleotide not encoded by VH. And so that's an N nucleotide. But then, of course, there's this match too, so maybe this is the recombination site, and that's just a mutation. Or actually, it could be that that is also an N nucleotide. It just happens by chance to match. And with this high a mutation rate, it's really hard to say. But it makes a big difference when you're trying to understand what the original form looked like. And so what we do is do the statistical analysis. We have a likelihood model based on this, this transition matrix showing the probability that A goes to D after time L. And we assume that the nucleotide positions are independent. This is what one typically does. And that the log of the overall likelihood is just the sum of the logs of the likelihoods of the individual positions. The nice thing about this, about approaching it from this way, is that we can immediately generalize to whole clones, to whole trees, right? So here's a case where I've got an antibody, and this is, I'm supposing that the observed state here is A, and I can ask what the ancestral state was, A, G, T, or C, and the probability function that I just showed you tells me what the probability is, what the likelihood is for any of these choices. Similarly here, I've observed A, G, A, and I hypothesize that this is the appropriate tree, and I can ask what is the likelihood for A, G, T, or C at the ancestral state here. And it's the ability to do this with clones that is so important. So now, for those of you who have some background in bioinformatics and you've done Waterman-type sequence alignments, what we can do now is we can use linear programming the way is done for Waterman-type sequence alignments, only now we use our likelihood function to generate the appropriate scores. So the alignment score for aligning X against D is just the log of the likelihood. We can similarly have a insertion score and a deletion score all based on exactly the same likelihood. And then the only thing that requires a little bit of additional thought is what you use for an N nucleotide score. But the point is that you can just use regular old dynamic programming as long as you make the appropriate substitutions. And so here's the overall algorithm. We have to align the sequences. We assume some initial tree. It doesn't really matter what we assume to begin with. In fact, what we typically do is assume a palm tree. So there's the unmutated ancestor, then a trunk up to the common ancestor to all the observed antibodies, and then just a star out from there. We compute the likelihood for each nucleotide at each position of the alignment, and then we align every one of the V gene segments to this likelihood. And then similarly for JH and DH, we compute the marginal probabilities and normalize by summing over all gene segments and recombination positions. So essentially what we're doing is we are aligning our tree against all possible ancestral states. It's a Bayesian computation, and we're literally looking at all possible ancestral combinations, but taking advantage of conditional independence of the gene segments, so we don't actually literally have to go through a billion sequences. Then we repeat the whole process after inferring a new tree. So we started with an arbitrary tree. Now we have our best guess for the true ancestor, and we can now infer a tree and repeat the entire process with a different tree. If we come up with a different tree the second time around, we just keep repeating until it converges. I've never had to go more than one additional time through. So using this, we can infer the unmutated ancestor. So let's look at the performance of the algorithm. How well does it actually work? So to test this, what I did was I looked at the 37,000 human heavy chains that one can find in GenBank, and I poured through all 37,000 looking for genes that appeared to be clonally related to each other. And there are actually several very large clones in GenBank, and one of them contains 84 sequences. And I put discovered here in quotation marks because I discovered this clone much the same way Columbus discovered America. That is, I found out that this is a clone. Then when I went back and I looked at the paper from which it came, of course, they recognized full well that it was a clone, and that's what the paper was all about. So I could have saved a little time by just reading papers, but that never is a good idea. Catherine will recognize that physicists would prefer to do the computations themselves, because 2 days of computation might actually save 2 hours in the library. So what we're going to do is we're going to use the entire 84 sequences to infer the ancestor, and then we're going to subsample smaller subsamples and use those to infer the ancestor. And we're going to do two things. We're going to estimate estimate the error frequency, because we're doing a full Bayesian analysis, which means that not only do I get the best guess, but I get to see what the uncertainty is at each position. And then we're going to actually compare the various inferred ancestors to each other. And we're going to compare what we get in those cases. So here's what we get. The colors of the dots indicate the sample size. So the green dots are 10 subsamples of size 27. The red dots are 10 subsamples of size 9, purple size 3, orange size 1. And this is the scatter plot. This is the error, the total error, the sum of the error over all positions estimated for each case. And this is the observed number of mismatches between each individual ancestor and the ancestor inferred from the full data set. And so we see that the estimated total error is nicely correlated with the observed number of mismatches. And in fact, the behavior is quite acceptable. And you can, stare at it a little bit longer, but it works out. So we feel comfortable that not only are we inferring the ancestor well, but we also have a very good estimate of the uncertainty. So let's put this to use. Let's actually use this to reconstruct the clonal history of an anti-influenza hemagglutinin clone. So now I have to introduce a wonderful technology that was developed by one group and adopted by Larry Liao in Bart Haynes' group. And this is a recombinant method for isolating and then synthesizing antibodies. In the old days, in order to get monoclonal antibodies, we had to make B-cell hybridomas, which is a long and error-prone, well, not error-prone, inefficient procedure. Here, what they do is they take blood from patients from whom they want to isolate antibodies, and they use flow cytometry to sort out plasma cells. They then distribute these plasma cells into multi-well plates, one cell per well, and then they use a multiplex RT-PCR to amplify the immunoglobulin heavy and light chain gene pairs. So they use multiplex PCR. They have multiple primers that are capable of amplifying any V heavy or heavy chain, and primers that are capable of amplifying any light chain. And in so doing, they are able to isolate the heavy chain, light chain pairs. Then they send that information to us for analysis of the sequences, and they make transfection constructs where they can now make very large quantities of these antibodies in cells that are specially made to do that. Once we have that antibody, we can now look at their properties, binding and neutralization, and so forth. So let's apply this to a specific case. We call it clone 2569. And in this particular case, this is a study that was done in England. Volunteers who all turned out to be Australian for a reason that I don't quite understand. I don't know. They voluntarily inhaled influenza, and 7 days after they inhaled the influenza, they gave blood samples, and our colleagues went through the procedures that I just outlined and isolated scores of antibodies from these patients. So what I did was then just go through all of the antibodies that they'd isolated and determine which ones belonged together within clones, which ones seemed to be clonally related. And there's one that produced a clone of size 5, and we look and we can build our phylogram and find that there are altogether among these 57 non-synonymous mutations and 31 synonymous mutations occurring along the branches of this tree. So it's a fairly well developed clone. So this is the inferred phylogram. 05, 02, 01, 03, 04. These are the observed antibodies, observed heavy chain, light chain pairs. A is the ancestor, is actually the best guess at the ancestor by the inference methods that I just described. And in addition, we can infer the intermediates through which we believe that the clonal history must have passed. So we infer the DNA sequences for these guys and also have the DNA sequences for these. But of course, the great thing about this recombinant technology, which you can't do if you're doing hybridomas, is that once I've inferred what these are, once I have the DNA sequence, I can make transfection constructs for those too. I don't need to actually isolate antibodies. I can just make the antibodies. And so that's what we did. We inferred these guys, we made antibodies for the ancestor and for all the intermediates, and then we measured the dissociation constant for each of these monoclonal antibodies. And this is the log base 10 of the dissociation constant. That's the inverse of the affinity. And so the ancestor starts up here. This is actually about 1 micromolar. And it the dissociation constant then decreases quite uniformly over evolutionary distance. So what we're doing here is you just project from this phylogram, these are the estimated branch lengths. You project down from here, I4 to I4, O3 to O3, and so forth, and you see that the affinity maturation appears to be occurring very uniformly and very consistently over time. So we were very excited to be able to do this, to study the clonal history by reverting these antibody genes. And I don't know if I'm going to be able to get this to play or not. Okay. This is just a little bit of fun. Kevin Weehee is a structural bioinformatician, and what he did was he took the trajectory along one of the paths from the ancestor through the intermediates into the observed antibody, and just predicted what the changes would be. We don't have a crystal structure, and so we don't really know what they're going to be. But he just thought, let's just see what typical molecular dynamic simulation would give you. And so what we're going to see is a set of changes through which Kevin predicts that the molecule might have gone. So again, this is not intended to say here's what happened and why, but it points out that once we are able to do these crystals, and that is indeed, it's actually happening right now with Steve Harrison's group, we will be able to see the stages through which these antibodies pass. So I would like to talk now about broadly neutralizing antibodies in a little bit more detail. This is really what we're truly after. Broadly neutralizing antibodies both against influenza and against HIV. Now, broadly neutralizing implies two things. It implies neutralizing, and it implies broadly. And I don't mean to belabor a point, but I just want to be careful about what it is I actually mean. So neutralizing means that an antibody is capable of preventing infection of a cell. So that it prevents the virus from entering the target cell. That's neutralizing. Broadly means that the antibody is capable of preventing viral entry even against a large number of different strains of the virus. And this is really key. It's key in HIV because as I've already mentioned, and as you're all very well aware, HIV mutates so rapidly that as soon as an antibody response comes up, if a virus has a mutation available to it to escape, it will, and it will do so within days. In flu, it's also very important because, as you know, the flu changes from year to year, and we have to reformulate the flu vaccine every year, and it's a guessing game. And if we're wrong, the vaccine is not very effective. So if one can generate broadly neutralizing antibodies by vaccination, then we have made a very substantial improvement in vaccine design. So there are several broadly neutralizing antibodies against HIV that have been isolated over the last 30 years. Not very many of them. They are very rare. And up until 5 years ago, there were only 5 of them that had been isolated. But because of the intense interest and the development of new technologies that are capable of doing these kinds of isolations, several more have been isolated over the past 5 years. And so these are all the ones that are known. And I want to point out one thing in particular. This is the percentage or the frequency of mutation in these antibodies. So the lowest we see here is 9.3%, and that's not a particularly potent broadly neutralizing antibody, by the way. And then the most mutated are 36% mutated. Now, Steve is impressed because Steve knows what these frequencies mean. For the rest of you, let me just show you a little background. So I mentioned the 37,000 human heavy chains in GenBank. So what I did, I just took all the heavy chains and I put them through this analysis and look at the mutation frequency in this collection. So nearly half of them have none or one or two, very, very small number of mutations. It drops off dramatically after about 8%, and by the time you're up to 16%, it's really hard to find any at all. And now compare it to here with our broadly neutralizing anti-HIV antibodies. Almost every one of these lies way out here in the tail. So for reasons that are not yet clear, almost all of the broadly neutralizing antibodies have accumulated very, very large numbers of mutations. So affinity maturation is without doubt working here. So let's apply our methods to an anti-HIV clone. So this particular... Yeah, sure, Steve.
Do they know for those individuals, do they genotype them so you know they're somatic and not polymorphic segments?
Which one? Oh, you mean these broadly neutralizing antibodies. Some, yes, but not all of them. So you're absolutely right. Some of the mutations could be due to allelic differences that have not been detected. This clone was isolated along with many other antibodies from a patient on day 20 post-transmission. This patient knew exactly the day on which he was infected because it was the day on which he engaged in the behavior that led to his becoming infected. And so we know that this is exactly 20 days post-infection. This clone is actually very large. There are 51 members that were individually isolated, among them just 17 unique ones, but it's still quite large. And the least mutated heavy chain light chain pair has 15 mutation, and the most mutated has 34. So again, this is a mature clone. Now, I want you to think about that too. This is 20 days post-infection, and the most mutated member has accumulated 34 mutations. Think about that, and then I'll come back to that. So this is the phylogram for that clone, and we were not able to make all of the intermediates because even though we are capable of doing it, it's still expensive. So we inferred the ancestor and several of the intermediates and measured the effective affinity, or in this particular case, again, it's the dissociation constant. And so we have the affinity of the unmutated common ancestor, what we believe the unmutated original rearrangement looked like. The dissociation constant is greater than 100, and this should be micromolar, not nanomolar. Greater than 100 micromolar. Sorry for all the typos on this slide. On all my slides, I mean. 100 micromolar. Now, what's significant about that is that 100 micromolar is very likely not capable of activating a B cell. So it suggests that the original antigen, the antigen that caused the activation of the ancestor and its proliferation, is not HIV. It's probably something else. And in fact, it's not until we get past this intermediate, which is also very low affinity, that we get to 65 nanomolar, which is now respectable. So this guy has a very distinct anti-HIV capability, 52, 34, 14. By the time we get out to the actual observed antibodies, this is a very high affinity antibody indeed, about 1 nanomolar, and that's typical of a high affinity, high specificity antibody. So, it looks as if we started with an antibody and a B cell that reacted against something else and expanded, And it was only later on in the process that HIV took over and picked up sort of in the middle of affinity maturation and drove it the rest of the way, so to speak. And this is also consistent. Oh yes, Tim.
With these trees, those represent probable ancestors all the way back to the track.
Yes, that's correct.
But because you could have some reversion mutations, you could have sequential mutations at the same position, presumably there's some uncertainty about whether the sequence you're inferring is the actual one that was on the history.
That's exactly right.
So do you have an estimate for how likely each of those ancestors is?
Yes, absolutely. Yeah, that's why they hired me in the first place, was because I could give them estimates of the uncertainty. Although, frankly, once I told them what the uncertainty was, they wished they hadn't hired me. Yeah, no, you're absolutely right. And so I would take any individual one of these reconstructions with a grain of salt, but we've now done dozens of these things.
But I was just wondering how big the grain of salt was.
Oh, so a reconstruction like this is typically going to have uncertainty in 3 or 4 nucleotide positions that can be 20 to 40% uncertain. So that means that it's very likely that that exact sequence is not correct. But the number of substitutions is likely to be small, and so we think that by looking at large numbers of these reconstructions, we're getting a reasonable understanding of what's going on. So what we think happened is that there was an antigen that elicited this clone to start with, and then HIV came along and by chance latched on to an already developed clone and then drove it the rest of the way through to the high affinity state that it has now. And in fact, we have now looked at several dozen similar clonal histories. This is the largest of them, but we've looked at many others as well. And this is the rule rather than the exception. So the ancestor is very often, majority of the time, not all the time, but the majority of the time, so low affinity that it's unlikely to have been elicited by HIV, and that it's only later on in the clonal history that HIV becomes capable of driving the rest of the expansion. I also want to mention that this is consistent with the high frequency of mutations that we're seeing in this clone. 20 days after infection is only time enough for one germinal center reaction. And that means that if we think that this was elicited by HIV, then all of these mutations would have had to accumulate over just 20 days, and that's very, very unlikely. Yes, yes. This is the autologous HIV. This is the strain that was transmitted to this individual.
Was it inferred? You inferred it?
No, it was isolated.
At 20 days?
The HIV was isolated at 20 days. The virus.
I mean, how do you know that early strain isn't attacking? I mean, HIV just mutated away. Do you know what happened to it?
Well, so it is possible that HIV mutated away, but the isolated virus at day 20 was not highly diversified. So what are the insights that we take away from clone 558 and the additional clones that we've studied afterwards? We believe that the clone pre-existed at the time of infection. The founder, unmutated common ancestor, is not reactive with the antigen to which it will eventually become reactive. And the clone, therefore, was elicited by some other antigen and then co-opted into the anti-HIV response. And as I have emphasized, this pattern is the rule rather than the exception. And so this observation and a lot of head scratching has led Bart Haynes's group, including Bart and Garnet Kelso, Steve Harrison, and myself, to put forward a new method for eliciting broadly neutralizing antibodies, particularly against HIV. Okay, well, this will have to wait till next year, so come to Boston. Very, very briefly, we isolate antibodies, we infer the ancestry, and we determine what the unmutated ancestor must have been. We then, and this is the hard part, this is why a high-power guy like Steve Harrison is involved, we then have to determine what the original antigen was. And we do this by a combination of experimental methods and computational methods, and vaccinate with the original antigen so that we can expand the pool of precursors. And then once we've got the expanded pool of precursors, we then boost with additional antigens attempting to drive us toward the antibodies that we seek. And let me just say that the first test of this is now going on in macaques, and we're all anxiously awaiting the first results, which we expect to get in the next couple of weeks. And so with that, I'm going to end, and you won't get to hear about germinal center modeling, but that's okay. Those of you who are in the summer school managed to see a little bit of that anyway. So thank you very much.